Okay. All right. Well, thanks everybody for joining. This session was put together by myself. I am now an employee of Slack since May, working the LCLS data systems group, along with Minhuan Lee is the other kind of like lead organizer. He's a graduate student in Duca Hextra's lab at Harvard. Say anything about your research or role?
Yeah, I'm working on like how to use like generative models to incorporate this like experimental information to do the stuff. And I'm finishing my PhD deploying the Flatiron Institute afterwards.
And we couldn't have done it without the support of Fred, who's also in the LCLS data systems. Knows the landscape around Slack a lot better than I do, and can smooth over a lot of connections. But yeah, so I just came up with the title for this workshop, and we called it "Building a General Inference Engine for Chemical Dynamics." It's quite like a somewhat provocative, but also quite vague title, and I hope that throughout the course of the day we can unpack what I meant by it. And to that point, I need to cop to the fact that I use the term chemical, but really I'm a biologist by training. And, in particular, my background is molecular biophysics. That was my PhD. I've been doing structural biology for many, many years now. And so when I think of chemical dynamics, it's usually this sort of thing. It's usually, I'm thinking in the context of an enzyme or another proteinaceous molecule that interests me as a biologist. Which is not to say that this session should be in any way biased against more general chemical questions, catalysis and so forth, material science, what have you. But that this is definitely what we were thinking about when we put together the speaker list. And to that end, I think it's a really exciting time to be in the field of structural biology. There's a lot of interesting things going on, both computationally and experimentally, methodologically. Right. We have finally, after decades of trying, kind of cracked the protein sequence to structure prediction problem in a spectacular way. And it's really transforming the landscape of biology and what biologists do day to day. That's one really exciting thing in the space. But also the experimental methods that we use to determine structures are getting really advanced in their capabilities, right? We've seen the first sort of couple of situations where cryo-EM is at proper atomic resolution. It's unclear how much further we can push it, but we're starting to see these stunning maps for very interesting proteins like apoferritin. That's a joke, you can laugh. And more excitingly, I think we're starting to see in situ structure determination with tomography. I really like this example. We're at about 10 angstrom inside of a cell, this really complicated protein structure that's spanning the bacterial periplasm. In crystallography land, we have got really, really bright light sources, right? The premier light source in the United States, the advanced photon source, is just coming off of an upgrade cycle, and it's getting a boost in flux by about 100-fold. So this is really going to be transformative in terms of the sorts of experiments that you can do at a synchrotron. Of course, we're on Slack's campus today, and we have the premier free electron laser in the world here, which delivers very, very intense, very fast X-ray pulses with which you can do some very interesting biology. To go with these advanced light sources, we've developed as a community advanced sample delivery techniques that can get a lot of samples into the beam quickly, so we can do damage-free observations of radiation-sensitive proteins and do really intricate time-resolved work in enzyme dynamics. So moving from this world where we had to grow very large single crystals to being able to study microcrystals, more marginal material that's in some cases easier to produce for these challenging systems, and study them in a time-resolved manner. Of course, when you talk about dynamics, crystallography is really kind of the new kid on the block compared to NMR. It's a very established suite of experiments in this field addressing, anywhere along the decades of biologically relevant timescales, right? And furthermore, we're starting to do time-resolved experiments in the realm of solution scattering as well. We have Tom Grant here today, who's going to tell us about how we can push the resolution, I think, of solution scattering, and make some really exciting advances, increase the information content of those data, which are they're closer to the native environment in some sense that a protein experiences in the cell, and in some cases easier to acquire as well. So it's very exciting stuff. Also leveraging the advanced light sources that we have. So what we thought as organizers was this is all setting the stage for moving to the next stage of structural biology, where we're moving from solving a single static structure of a protein of interest to looking at dynamics. And I mean dynamics in the sort of equilibrium sense, a single protein diffusing on an energy landscape, and as well as the chemical sense, where you're carrying out a reaction, bonds are breaking, things are changing in the sample. And the hope is that we can take these sorts of studies that you see here in this DHFR example from Mike Sawaya, and take them from a place where they take somebody's PhD or maybe a handful of PhDs to get these sorts of results and compress them into a more routine, approachable sort of measurement. And I think that, at least in my own work, I feel the thing that is missing, so we have all of these great experimental tools now, is the inference pipeline to go from data to a picture like this. So just to sort of frame the discussion, wanted to talk about where we're at in structural biology and where I think we'll be going to move into this more dynamic space. This is sort of the picture of structural biology as think about it like 10 years ago, right? You have these three major structural determination techniques. They're kind of siloed. And we have individual analysis tools for each of them. Each of them can take their domain-specific data, produce a structure. We have validation tools that are separate for every sort of experimental modality. And then we have a database where they can all be deposited together with their own sort of appropriate metadata. What's happened more recently, and this is really sort of the amazing outgrowth of having the protein data bank and having close to 200,000 X-ray structures in it, is that publicly available database has been combined with advances in deep learning to give us these amazing structure prediction models, right? And critically, these wouldn't have been possible without sort of conditioning, augmenting information from other sources. So in this case, of like AlphaFold 2 and ESMFold and these very successful structure prediction models, that info came in the form of DNA sequencing data. And I think the amazing thing that has grown out of this, and something that I hope we can really appreciate as a community and leverage going forward as we want to move into the dynamic space, is that the existence of these structure prediction algorithms had a this really interesting feedback loop, which I actually like to think of as cooperation between an AI agent and a human scientific community. And this happened the moment that the Phoenix developers decided to integrate AlphaFold 2 into their molecular replacement software, right? For those of you who aren't crystallographers by background, there's this problem in crystallography where you have to get initial phase estimates for your experimental data. And the way that's done by and large now is starting from an AlphaFold prediction. And that has been so enshrined that it's actually part of our core utilities for solving these structures. So what this sets up is a situation where a structure prediction model can make a prediction about a structure that it's completely naive to, and then an experimentalist can go ahead and use that to try and solve their structure. And that feeds back into the deposition pipeline and will eventually end up back in the model's training data set. So there's a feedback loop here where the model actually gets to guide exploration of structure space. Okay? I think it's mind-blowing. I think that would be, that was certainly something that was unimaginable when I started in structural biology. But I won't, you can take issue with how I've drawn this, but this is how I conceptualize the landscape at the moment. It's good to talk about it throughout the day. But my question now is, how is this going to be augmented over time? What is the landscape going to look like in 10 to 15 years? And I think that, it doesn't have to be that different. The bones can still be the same, but I think that we should start making some really key fundamental choices right now. For instance, I don't think that we should keep maintaining separate domain-specific analysis tools. I think we should start taking, what I would call multimodal data more seriously, what I think Andre would call integrative more seriously, and design the next generation of structure interpretation tools with this in mind, that we're going to incorporate data from multiple sources, probably most of the time when we deposit a structure. I think that we should imbue these models with the ability to understand dynamics in some sense, either at equilibrium, so diffusing on a sort of static energy landscape, so recovering an ensemble in that case instead of a single point estimate of the structure, but also incorporating time resolved information like we're getting from these advanced light sources. And we'll probably hear more about this today, but I think we need to take very seriously how we're going to deposit and validate these sorts of data as well. And it's going to be different from what we have at the moment. This is all fine. The models will change as we grow as an inference discipline, but hopefully we'll keep that aspect of the architecture the same, but we'll start to take seriously predicting ensembles and predicting dynamics instead of maximum likelihood estimates of a single structure as well. And of course, try to preserve this feedback loop concept. Do you have something to say?
Yeah, just today we're going to focus on this two red box stuff, about people from different field expertise, trying to talk about the analysis tool at the moment, and we can go to further discussion about what is the possible thing we can do to extend to be multimodal.
That's right. Yeah. So what we have done with this workshop is to try to get together who we think are some of the finest representatives from each of these three communities, and from solution scattering as well. And start to think about how we can build a general inference tool rather than having a separate one for each kind of domain. we're doing well on time. So the schedule, I think is accurate on the conference website.
Yeah.
Yeah. Okay, good. So we've intentionally made the choice to make it quite leisurely. Our hope is that there's going to be a lot of discussion throughout the day. And this morning, we'll hear first from Andre Shelley from UCSF, and then from Piotr Bukowski from ETH about NMR. I think Andre, we didn't get a talk title from you, but I assume you're going to talk about like integrative structural biology. Perfect, or you can. Well, sorry about that. No, that's okay. I'm not trying to shame you. I just want to know where we're going. So yeah, then we'll have a relaxing coffee break. We'll come back. We'll hear from Alizia Fedini, who's at Cambridge in Randy Reed's group, about how we can incorporate AlphaFold already into interpreting crystallography data. And then we'll hear finally from Pilar Cosio from Flatiron about cryo-EM. And then we'll have a lunch break and come back and hear more excellent science. So I just put a list here of sort of the key questions I want people to be thinking about throughout the day. And I think, every speaker on this list has something to contribute to all or at least most of these questions. But to me, these are some of the key things we need to answer as we're looking toward the future. So start off like, what kinds of inference machinery do we want to use? Do we want to keep doing like maximum likelihood inference for the rest of eternity, or do we want to be more Bayesian about things? Do we want to do math estimation, or do we want to use variational inference? In the future, will we be so like flush with computational power that we'll be able to fit everything by MCMC? I don't know. And are we going to start incorporating some of these more advanced generative models that are have been so transformative in the AI space? So diffusion models and so forth. What kinds of strategies for conditioning on external information can we use? Like, can we use more Bayesian approaches with sort of interpretable prior distributions, or do we want to be looking at kind of like the cross attention sorts of approaches that are used in text to image models, which are a little bit more black box, but obviously very powerful. And then like a key question for each presenter, because we have a diverse set of fields here, is what do you care about in your chosen discipline? Like, what are the experimental observables you have, and what can we calculate them from? What sort of parameterizations of protein structure and dynamics do we need to use to make sure that we can actually fit your data when it comes time to do so? Should we be learning atomic coordinates? Should we be learning density maps of various kinds? How do we want to parameterize this thing? Or should it all be done in some like completely abstract latent space that's high dimensional, and we don't like directly interpret it, but we have decoder modules that can understand it? And then what lessons from your own field can we bring about interpreting dynamics? My own field of crystallography is relatively new to this, but I think we'll hear great examples of how you can infer these sorts of at least thermodynamic information of ensembles from crystalline samples even. And yeah, what insights are from your domain are transferable to the general situation? And critically is like, what infrastructure do we need? What sorts of data repositories do we need? What sorts of validation do we need? All these kinds of questions. I hope we can, make some progress on throughout the day. And I think with that, I can hand over the floor to our first speaker.